All right, good afternoon, morning, evening, whenever you're watching this, Anatomy Physiology, but this is the presentation of the muscular systems if you'd like to watch it. So when we're going to talk about the muscular system, we're going to kind of review back to histology when we talked about the three types of muscles. We've got skeletal, smooth, and cardiac. The skeletal muscle is what we're going to be focusing on in this section. So that skeletal muscle will be this one right here. This is the one that we're going to focus on in this particular lesson because it's straight involuntary. We do have smooth and we do have the cardiac, and we will be focusing on those a little bit later. So let me just kind of delete those. So the skeletal is the one that we're going to be focusing on, but the big thing there is the focus on the skeletal muscle. So muscles and muscle fiber structures, this is what you're going to be doing probably Friday, is that muscles are composed of many fibers that are arranged in bundles called fascicles. So we're going to be doing a lab with Twizzlers. And so the fibers would be like a single Twizzler and then a whole bunch of Twizzlers wrapped up as a fascicle. So we'll talk about that. So again, a little kind of graphic picture here, but individual muscles are separated by fascia, which also forms a tendon. And so on your heel bone, there's strained plantar fascia. And so we talked about plantar fasciitis, but the individual muscles have this fascia that's covering around them. And so we'll be, again, modeling this, but that's kind of give you a look at that foot there. So I would pause this video and maybe write these down. But so epimysium is the outermost layer, surrounds the entire muscle. The key part here, I think, is this term epi. If you remember, if it's going to let me write, like epidermis outside. Perimysium, it's separates, surrounds the fascicles, and then the endo surrounds the individual fibers. And so what you're going to want to do is, when we're going through this thing about epi, para, and endo. So the epimysium, again, is the outer. Para is going to separate those fascicles, and the endo is what's going to round the fiber. And again, you're going to be building a model there. So there's an image there. This of using straws and stuff, but we don't want to use straws. Oops, let me get back to my slide here. So the muscle layers. So again, epi is the outside. There's your paramecium and endomecium, and then there's your muscle fibers. So we're going to be building models later. And so again, here's another image of it. So you may want to pause and draw that. So you can see the endo is the innermost black, the para, and then the epi. So muscle cells, when we talk about muscle cells, there's a couple things we need to talk about. Sarcolemma, that's a muscle fiber membrane. So if you think of like a cell membrane, sarcoplasm is the inner material which surrounds the fibers, like in cytoplasm. The sarcoplasmic reticulum transport, and specifically it's going to transport calcium. Let me see if I'll be able to write calcium on here because I got to remember calcium, and that's not what I wanted. So I don't know how this writing thing is not going to work. So anyways, we'll do some board, but it transports calcium, and calcium is important for muscle contraction. And then the myofibrils, those are individual muscle fibers within the sarcoplasm. And again, I will be showing you a picture here of this later. So here we go. You've got your nucleus. So this is a muscle fiber. There's your sarcolemma, which is essentially the cell membrane. You've got the mitochondria in there because you need energy, right? The sarcoplasm is all that substance in between, and there's your myofibril. So there is kind of just the structure. Again, you're going to be building this out of Twizzlers. So your myofibrils are made of two types of material. You've got actin and myosin. Actin is thin filament, and then myosin is thick. And so again, the myofibril, and again, you're going to have to kind of learn these sequences here, but that's what they're made up of. And so the actin and myosin are just the very thin filaments. Think like fishing line almost as in terms of a filament. And then those filaments, actin and myosin, are even smaller. And we have these things called dark and light bands. So an A band is the dark band, and that's the thick band, which is the myosin. And the I band is the light band, and that's the thin, and that's the actin. And so if you look here on that picture there, you've got dark A, light I. And so these are just the structures there. And so again, you may want to draw this in. Whoops. And so again, this is kind of going from largest to smallest. Again, I'm going to try to draw in here if it'll let me. So here's the muscle fiber. Nope, it's not going to let me do it. There's your muscle fiber. We talked about the mitochondria. There's your sarcolemma. So what this is showing you is here, we take this piece, we make it much smaller, right? We're going to draw our arrow down, and this is just showing you this piece zoomed in. And so you've got your thin, which is the actin. You have your thick, which is the myosin, and then you have these different zones, which I may or may not go over. But this is just kind of showing you these different structures here. And I got to figure out a way. I wish I could delete all at once, but I haven't quite figured that out yet. So we'll get there. So let me just delete these as we go. All right. So, and again, we're going to be building models, and hopefully they'll work out. So, pen. does not want to stop working. All right. All right. So here's a sarcomere that's contractual unit. As you can see, you got these Z lines, and everything moves towards the middle. And that's the key point, is that when a muscle contracts, it gets smaller, so things move towards the middle. So here's again some muscle to scale, and just kind of showing new structures again, going from largest to smallest. You can see the bone, the tendon, as we break it down smaller and smaller. So here, it's important to remember this hierarchy. It goes muscle is composed of fascicles, then the fascicles are composed of myofibrils, then the myofibrils are composed of myofilaments, and then the myofilaments are made up of actin and myosin. So again, I would pause this and jot this down and add bundles and fibers and filaments. We're going from large to small here. But again, the key point is everything moves towards the middle. And so we talk about muscle contraction, we got to talk about the neuromuscular junction. So if we take that term neuromuscular and break it up into two words, and again, I don't know if it's going to let me do this, just go through, divide that in half. Neuro meaning nervous system, muscular. So this is how the nerves and the muscles interact, because that's essentially what causes the contraction. We haven't talked about nervous system yet, but this is the nerve part here. These are neurotransmitters, so think like acetylcholine, and here's the receptor on the let me see if I can clear everything at once. Nope. That's the on the muscle, excuse me, that's where the neurotransmitters will be picked up. So this may be not as well as I would have liked. So neuromuscular junctions, where a nerve and muscle fiber come together. You have a motor end plate. That's where the muscle and neuron communicate. You've got the synapse, and then you have the synaptic vesicle. And so these are the neurotransmitters are things like acetylcholine. And so what I'm probably gonna have you do in class, we're gonna have you draw this and label it. So 1's the neuron, 2's the sarcolemma, which is this, or the motor end plate, which is the neuron there. Excuse me. 3 are vesicles. 4 is the synapse. 5 is mitochondria. 6 is receptor, and 7 is acetylcholine. And the acetylcholine is the name of that neurotransmitter which causes the contraction. So I would pause the video and draw this. And so the neurotransmitter that crosses the gap against acetylcholine. So acetylcholine is broken down by cholinesterase, which is an enzyme. We know it's an enzyme because it's ends in ACE. And so there's often types of disorders where the acetylcholine is not broken down and just causes that muscle to constantly contract. And so again, the neurotransmitter that crosses the gap against acetylcholine, and this is what activates the muscle. So. And there's probably going to be a video here. And so I'm going to probably, I'm trying to see where we're at here, other term, threshold. All right, I'm going to stop, excuse me. So the sliding filament theory is where I'm going to stop, because you're going to watch a video. But the sliding filament theory is this idea of how muscle contracts. And the contraction of muscle occurs as the thin filament slide past the thick. And so what is needed for this to occur is ATP, calcium, myosin actin, acetylcholine, and cholinesterase. So I think this is my plan. This is just going to be, we're just going to call this muscle movements part 1. I'm going to do this in class, but yeah, because I don't want to overwhelm you, I'm going to keep this to about less than 10 minutes if I can. So this is about half the slides. All right. So I hope this makes a little bit of sense. This is just to help you out. Introduction, pause, draw images or whatnot. I don't think me drawing on the screen is going to be very helpful. So anyways, this is me sign off from room 102. Have a good day. Hopefully you found this helpful, and I will see you in a later video cast.